Welcome to the webinar on how to manage spider mites in tomato with predatory mites. This is the fourth and final webinar in a series about biological control of vegetables in the Southeast as part of a NIFA OREI funded project. So today I'd like to welcome back Tom Bilbo and Jim Walgenbach. Tom is an assistant professor of vegetable and strawberry entomology at Clemson University, and Jim is a professor and extension entomology specialist at North Carolina State University. So he'll be going first. So with that, Jim, I'm just going to hand over everything to you and turn off my video.
Okay, thank you for the introduction, Alice. It's a pleasure to join everyone here on a nice afternoon, at least it's afternoon here in eastern part of the United States or in North Carolina. Today, it's going to be Tom and myself going through this information. This is kind of a long history. This project is like in its fourth and final year, and it's kind of interesting. I think when we first started this project, Tom was still in graduate school, and here we are 4 years later. And now he's the project director of it. So he knows how to move up the ladder quite quickly. So he can possibly explain why he is the project director. But in any event, our part of this was management of spider mites in tomatoes. And what I'm going to talk a little bit about today is just a little bit of the background information on mites in tomato systems, some of the factors that contribute to them being outbreak pests, and a little bit about biological control. But Tom is going to be talking primarily about the biocontrol aspects in terms of how you can go about manipulating a key predator to enhance biocontrol in tomato systems. Well, there's about 5 different species of mites that potentially are a problem on tomatoes. And they are in 3 different families. The Trichinidae which are the spider mites, consist of the 2-spotted spider mite, which is Trichomachus urticae. Then there's also the carmine mite and the tomato red spider mite. Then there are 2 other species. One is the tomato russet mite and the broad mite that are very small and not really visible with the naked eye, opposed to the spider mites, which are naked, they're naked, but they're also visible with the naked eye. Both the tomato russet mite and the broad mite are very small individuals, and you really need a microscope to be able to see those. The spider mites, as indicated, there's 3 of them. The 2-spotted spider mite and the carmine mite, they're very similar. They have a very similar life history. All 3 of these actually do. And they're very similar. The spider mite, or the tomato spotted spider mite and carmine mite, can be oftentimes occur simultaneously within fields. The spider mite is a worldwide pest that ranges from temperate to subtropical regions. Same thing with the carmine mite. It's usually less frequently encountered than the 2-spotted spider mite, but oftentimes later in the season when we really have much hotter conditions, it seems to appear a little bit more frequently. Then the tomato red spider mite is actually an invasive species that's originally from Brazil, and it was in the 1980s or so when it first started to disperse to other parts of the world. And it's more of a tropical area, or a tropical type of pest. In the United States, it's really only present in Florida and some of those southern Gulf Coast areas, as well as Southern California and Arizona as well. But so it has a much more limited distribution. And again, that is as you can see here, it's very similar to the 2-spotted in terms of its life history, but it is red in color, opposed to the 2-spotted, which is a more cream colored and has those characteristic 2 spots on each side of its abdomen. The other 2 species that I mentioned, the tomato rust mite and broad mite, I most frequently encounter those in greenhouse situations, but they do also occur in the field occasionally. Usually when we have pretty warm temperatures, the broad mites tends to be more frequently encountered in the field than the tomato russet mite. But the tomato russet mite really is a specialist of solanaceous crops, and when feeding on the leaves, you can see it can dry out and cause leaves to bronze and die quite quickly. The broad mite kind of has a different type of it. It causes the gnarling of that new growth oftentimes, so you get some misshapen leaves, and it's oftentimes a common pest on pepper as well. Both of these mites, though, they can feed on fruit and cause a russeting, as you can see here. Both of them can cause that, and that also occurs on other solanaceous plants that fruit that they're feeding on. But today we're going to focus on the two-spotted spider mite primarily because it's by far the most important of all of these mites in field settings. And as you can see here, this species, as well as most of the spider mites, they also are spinning or releasing the silk, and you get this webbing over the leaves when populations are very high. As you point out, once you get populations to the extent where you see webbing like you see in the right hand side of the screen, it's really difficult to get things under control at that period of time because they're so well protected within all of that webbing that it's very difficult to get a pesticide to them. And in a bit I'll show you why natural enemies oftentimes have a very difficult time navigating their way through there as well. But here you can see the bronzing on the tomato leaves, and obviously when you have a lot of bronzing, that's cutting down on the photosynthesis and increases the rate of respiration, so you're reducing yields in that manner, both directly and indirectly, because they can also feed on fruit and cause what's commonly referred to as gold fleck. They're one of a couple of different insects that can cause gold flecks. Thrips can also do that, and I've actually seen aphids do it when they're really high in number as well too. So that's the type of damage. Tom, we were looking for this picture before, and here it is. It magically appeared when I was least expecting it. This is kind of a transition picture here, though. This is in Western North Carolina near me. Some of the challenges to managing two-spotted spider mites relate to their life history. They have a very short generation time and high reproductive potential. Under warm conditions, they can complete a generation in less than a week during really hot, warm conditions. They have very limited mobility, and so when you have that combined with a very poor crop rotation, you have a localized population that's well adapted to tomato. I should point out that even though the two-spotted spider mite has a very broad host range, it takes a while to adapt to different host plants. And if you plant tomatoes in an area that's never had tomatoes, those might not be adapted to tomatoes. So the spider mites may not be a problem in that particular area. But where you grow potatoes over many, many years, the mites are very well adapted to tomato. And tomatoes is a pretty harsh chemical or a pretty harsh plant, which I'll talk about in a little bit. And again, very few natural enemies, at least in our system. And then there's a very low tolerance for damage, which consequently has led to a dependence on a lot of chemicals for managing them, which can complicate the situation. I just wanted to point out in terms of sampling. Sampling can be kind of difficult because they're going to occur all over the plant. Most frequently, they prefer the upper portion of the plant, the upper third of the plant, although not strictly, but populations are always going to be higher up there. And the way we usually sample them, and which is a very consistent sample method, is we don't look at the whole leaf, we just count a leaflet. And usually it's that terminal leaflet on one of the first mature leaves from the top of the plant. So you go to the top of the plant, and when you find your first mature leaf, which is usually about the third or fourth plant or leaf down from the top, from the apex part, it's a good sized leaf, and we just focus on that terminal leaflet just to be consistent. We usually take 10 leaflet samples, and if we sample in 3 to 5 locations per field, it usually gives a really good estimate of what's going on out there. It's important though. Oftentimes those infestations are coming from the edge of the field, so you usually want to begin sampling on the edge of the field rather than the interior of the field to detect those early infestations. Although that's not always the case, because the way tomatoes have a lot of hand labor, and when you have workers going from one field to the other, if they're just in a field that had a lot of mites in it, they may be carrying them on their clothes and things and move them into fields in that way as well. So this slide is in here just to demonstrate the low tolerance for damage with spider mites on tomatoes and how difficult it can be to keep pest populations below an economic injury level. So shown here in the black with the data points, that's a mite population on our research station. And you'll notice it's almost a logarithmic growth rate once they begin to increase between June and early July. They just grow very rapidly. And our threshold level, the economic injury level, which is really the amount of damage they inflict that is equivalent to the cost of managing them. So it's kind of a break-even point. Above that, populations above that, it's going to cost you money. Below that, it's not really a justification to spray a pesticide or implement control measures. So that red line is the economic injury level, but the economic threshold level is really needs to be a little bit below that economic injury level, because you need a reaction time once you detect a population. That's only about anywhere from 3 to 5 mites per leaflet. So, and then the economic injury level is between 8 and 10 mites per leaflet. So, pretty low population. Why are mites oftentimes a problem? Well, pesticides are often the cause behind the situation. In conventional production, there's several types of insecticides that can flare populations. Pyrethroids are most notorious for that. Some neonicotinoids can also cause flaring of populations. And then some miticides or fungicides, they can oftentimes result in increased populations as well. And I'll point out why here in a little bit. In organic production, pyrethrins are like such as Pyganic is one material. Azera has Pyganic in it as well. They're actually very closely related to pyrethroids, and they can flare mites as well. So relying on those type of products is probably not a good way of preventing mite populations in the organic systems. Those materials that can suppress populations, obviously many miticides and chemical miticides in conventional production, but in organic production, and Trust has some acaricidal activity, and so you can suppress mites a little bit with that. Sulfur is also a very good product for controlling mites, although that can be difficult sometimes to use, especially in very hot conditions. And then I just put this up there. This is kind of in a conventional systems of some of the different miticides that are used that can provide control. And then what I have shown here is the cumulative number of mite days. So in other words, how mite populations increase after an application. So the higher the number here, the less effective it is. And this black bar right here is the control where it's not been sprayed at all. And then obviously the most effective ones here were bifenazate, which is acromite, and abamectin, which is agromec. And then these others are kind of in between in terms of efficacy. But how about materials that may be available to us organically? This was a trial we did several years ago. We kind of evaluated it in a different manner, where we looked at how populations increased when these materials are sprayed at every generation. This was based on laboratory data, but you can see the materials we evaluated were Venerate, Grandivo, and then a neem oil material, and then Tritech, which is a a product that is also an oil that has insecticidal as well as fungicidal properties. And then as a control, we just kind of used acromite, which usually just blows mites away. So, but it's what quite evident here is the most effective of these materials was Grandivo. And so that's a product that fits into organic production systems. You can see the higher the rate, the more effective it was. The neem oil was intermediate, as well as the Tritech was intermediate, and Venerate was not nearly as effective as these other products. So something to keep in mind. So I'm going to move on to biological control here, the introduction of it just a little bit. And there are several different natural enemies that can help suppress mite populations. Some of our generalist predators are coccinellids, such as lady beetles, damsel bugs, big-eyed bugs, and Aureus insidiosus and anthracourids. So these are generalist predators that generally are not that effective in terms of suppressing spider mite populations because they can't keep up with them as quickly. And they're oftentimes more interested in other insects such as aphids. Aphids or thrips than they are spider mites. But there are some phytociids, which are predatory mites, which can be very effective or effective biological control agents of two-spotted spider mites. One thing that does does decrease their effectiveness in tomato systems is the trichome systems on tomatoes. And this is a picture. If you look at a tomato leaf or even a tomato stem, you can see a lot of trichomes on there, very hairy plants. And then when you look at those close up, you'll see these bulbulous trichomes. And when they're disrupted and broken, they release a chemical that becomes quite sticky, and the insects just cannot move around within that complex of trichomes, especially those bulbulous ones. So that really decreases the efficacy of predatory mites in this system. There is one exception, though. The other thing I wanted to mention, can be a very effective biological control, is an entomopathogen. It's the disease of mites, is a fungus called Neozygites floridana. And this, when under humid conditions, and in the southeastern United States, we have a lot of humid conditions, especially if you get a rain event, it can decimate a high population of mites very quickly. It's naturally occurring in the environment, and you can see here they get brownish in appearance. These are dead ones, and if you look at them under a scope very closely, you can see all those spores of the fungus. Unfortunately, that fungus is also very susceptible to the fungicides that are used in commercial production of tomatoes. Tomatoes have a lot of diseases. There's a lot of fungicides that are sprayed that basically eliminate this as a biocontrol tool. So it's not unusual to walk through a tomato field and see a lot of mites, and you move 10 yards away outside of that tomato field, and it's very difficult to find mites oftentimes because this Neozygites has eliminated the pest in that area. But we don't have that problem many times in organic tomatoes because we don't have fungicides that are suppressing those populations. So if it's a humid condition, which is also not good for diseases, this disease should take care of spider mites in many situations. Next, I want to move on to a different predator, a specialized predator. And this is in North Carolina, where we've been doing our work in Henderson County. That's where our main campus is, in Raleigh, which is in the middle of the state. I'm at a research station in the mountains. But we do a lot of research in the Piedmont area as well, where there's a big tomato producing area. The mountains have a cooler environment than the Piedmont area. Piedmont can get quite warm in the summertime, and it has a more mild winter. And in that area, about 15 years ago or so, if not a little bit more, we found this specialized predatory mite called Phytocilius persimilis. And this only feeds on spider mites. It doesn't feed on any other mite species. As a matter of fact, it won't even feed on some other spider mites. It's a specialist two-spotted spider mite. But it can do a very good job. It's very quick. It's a mobile pest, and it has a voracious appetite. Just wanted to show you, we did some studies several years ago where we were looking at how well it could overwinter in different areas. And we put these plants infested with mites and also containing persimilis up along in these little cages in the mountains, the Piedmont and the coastal plains area. And we found them we found them to show that they overwintered in the Piedmont and the coastal plains, but we did not catch them in the mountains here. So they don't survive as well in the mountains as they do in the Piedmont and coastal plains area. Again, it's a specialist predator, voracious feeder of mites. It has very rapid developmental rate, and it's well adapted to high temperatures and high humidity, which is the situation we have in the summertime in North Carolina, South Carolina, throughout the Southeast. And there's some evidence to suggest that they really need to adapt to tomatoes to be effective. But they can quickly adapt to tomatoes if they're reared for several generations on those plants. And I just wanted to point out one reason why there's such good predators against the two-spotted spider mite. This shows you how quickly the two-spotted spider mite will complete a life cycle on the left here at 59, 68, and 86 degrees, anywhere from about, as you would expect. The higher the temperature, the quicker development, about a week at an average temperature of 86, whereas a little over a month at about 60. But Phytocilius persimilis develops much more rapidly. So consequently, that population can increase more quickly than the spider mite population. That's what makes it such a good predator against that pest. And this just shows you quickly some data when we first started doing this work, where we had 3 different plantings of tomatoes, and this shows you the arrow on there is when a miticide or a caricide was applied, and then this shows you in the red is when Phytocilius persimilis appeared. And you can see early in the season, Phytocilius does not really show up very quickly. It takes them a while to become present in the system. But so that's why they're oftentimes more effective on later plantings than they are on those early plantings of tomatoes. There's a lag time in the buildup between the spider mites and the predators. Now, what really works good is if you happen to have a planting right next to an early planting, then you can get a head start on increasing your persimilis population. Then we were, to help reduce that lag time, we were doing some work where we were rearing Phytocilius ourselves. And Tom's going to go into this a little bit more, so I'm just going to quickly go through it. The only thing I want to point out is that we have to have one greenhouse where we reared 2 spotted spider mites alone, and then in a different area of the farm, we had a little makeshift house where we reared Phytocilius. You got to keep them separated, because if you get Phytocilius persimilis in your spider mite rearing house, you can't rear mites anymore. So I'm not going to go over this. Tom is going to point that out. But one thing I wanted to point out is the cost of releasing only persimilis to control 2-spotted spider mite is really cost prohibitive if you're releasing them at the recommended rate of 20,000 per acre. It's going to cost about $740 per hectare, compared to an acaricide, which in some instances you can get by with only $16 per hectare. What may be more common, though, in some of these other areas where they're applying 3 acaricides, and you're looking at about $250 to $300 per hectare. So there's a big difference in that cost of the Phytocilius persimilis. I think what Tom's going to talk about is how you can get around having to release them across the whole field. So Tom, how about if you take over?
Will do. Let's see. Thanks, Jim, for giving the background on spider mites. Let's go. Okay. Should be good. So I'm Tom Bilbo. Most of this work that I'll be speaking about, well, I'm currently at Clemson University as an assistant professor, but I did a lot of this work when I was with Jim for 3 years as a postdoc in North Carolina State in Western North Carolina. So I'll speak about sort of how we developed this Phytocelus persimilis system. And an overview is I'll talk about optimizing predator release strategies, some possible differences among predator strains, the importance of insecticide compatibility, rearing your own predators, and conservation of those resident populations that Jim was just talking about. And I'll touch a little bit on another predatory mite, Amblyseius swirskyii, some goals, but also sort of the research, a little bit of the research that was done and challenges for tomatoes specifically. Oh, there it goes. Okay. So kind of as Jim showed, these two figures here are showing what spider mite populations tend to look like in these tomato fields when there's no natural enemies present, and there often isn't because tomatoes are such a hostile environment for biological control. Unfortunately, they keep out a lot of insects, but a lot of those insects are the beneficial natural enemies we'd like. The spider mites have just adapted, and so they kind of end up being like a predator-free zone. And in these systems, you see spider mites can enter into a tomato fields. They get going. A farmer, conventional, often applies an acaricide, and that may knock them back for a short period of time, but they quickly rebound under hot, dry conditions and really take off. And this is some data we have showing the efficacy of miticides in commercial tomato fields. Over 2 years across a number of fields. And you can see what spider mite numbers look like before the miticide is applied. And you can see that 1 week after, there's a reduction. The miticides have done what you want them to do. But 2 weeks later, the reduction you have is pretty much eliminated and the spider mites have already bounced back. And so miticides can play a really important role, but oftentimes their effect is very transient. And when you have really high spider mite numbers in one field, it can actually be problematic and cause problems in adjacent fields if those spider mites really get out of hand. And it was an example we saw in North Carolina. Strawberries are grown in the springtime before tomatoes, and oftentimes in the same agro ecosystem. This is the farmer that had a big strawberry field right next to where they had their tomatoes. Those strawberries ended up having really high numbers of spider mites in them. And then the tomatoes went in, and then, probably not a coincidence, but the tomato section right next to the strawberries got absolutely decimated by spider mites. And so it's highly probable that those spider mites came from the really high numbers that were initially in strawberries. And so when you have a problem in one field, you don't want it causing problems in other fields. And that's the importance of having a good long-term seasonal management. And as Jim said, Phytocelus persimilis, it's a specialist predator. It really is only going to be where the spider mites are. So because it's a specialist, it's also the most effective spider mite predator of the predatory mites. And Jim touched on most of this, but you can buy it from companies. This has been commonly done in greenhouses for many decades, and we've been trying to adapt it into field scenarios. Right now, the only widespread use in fields are strawberries in California and Florida, and there the dynamics are very different than tomatoes. This is what a persimilis would look like under a 10x hand lens on a tomato leaf. And so you see it's bright red, it has an oblong teardrop shape, which actually makes it easier to identify than most predatory mites. Now, when trying to improve releasing predators like persimilis, it raises a number of questions about how to actually do this the most effective way. Where do you release them in a field? How much do you release? When do you release them? So maybe the surrounding fields could possibly be important if there are other crops with other spider mites. And also potentially the source of the predators, whether those are going to be tomato adapted, reared on tomatoes for multiple generations, like a lot of the research we've done, or those that have been bean adapted. So these are predators that have only lived their lives on bean plants for many generations. So here I'm going to walk through an example of an inoculative release approach using Persimilis in tomatoes. We used the benchmark rate that's recommended by companies at 20,000 per acre, but we were only doing this in a small section of a field. And so what we did is that we were scouting these fields weekly, looking for spider mites when they first appeared, and they tend to appear usually in one section first, and they're very aggregated to begin with. And so in that first section where they appear, we'd go in and we would release Persimilis. And so this would often be a 5 to 10% of the total area. And then we'd release the predators, and then we'd come back and we would check weekly and we'd look at how the predator-prey dynamics were changing. So this is kind of what it would look like. We would be rearing these predators, and then we bring them out into the field on whole tomato plants, which are at this point would have primarily predators, but it's still a few residual tomato spider mites hanging on, maybe providing a little initial food. This is what sampling in a field looks like in North Carolina. Pulling off a tomato leaflet there. So in this example here, we're looking at a 3-acre tomato field, and it's split into 11 groups. Each one is like 5 rows wide, and each group's about 1/10 of an acre. And so we sampled this field, and spider mites first appeared there in that group towards the end, group 2. And so we went into that section, released Persimilis at that 20,000 per acre rate, which was only 2,500 here. And then over the next 3 to 4 weeks is that area became basically a little Persimilis producing factory for the rest of the field. And those predators then spread and dispersed and then managed spider mites throughout the entire field. So it was an inoculation in this case. And these are the spider mite and the Persimilis numbers in that entire field throughout that season. So the black line are spider mites. The spider mites, they come in. The grower gets concerned. They actually applied acromite there. It held the spider mites back for a week, but then they really took off and started increasing quite rapidly. But by that time, the predators finally got established, caught up, and then completely eliminated spider mites from this field, so much that the predators then had to disperse and find food elsewhere. And I should note that we actually got the predators out a week later than would have been ideal in this case. So the spider mite curve there would have been much lower likely. And so it also, the question of making the system not only effective but economical so that it's actually adopted and used by growers, is that are there ways to extend the benefits of releasing predators like this? And so the answer is there's definitely potential there if you strategically position crops. And in one case that would be strategically positioning different tomato plantings, which are staggered throughout the season next to each other. And this would be an example here, this image of 2 tomato fields, a June planting next to a July planting. And so in that June planting, we did just like what I was showing before. We scouted that field and we looked for when spider mites first showed up. And then in that area, we released the predators. And then we let them, carry out the same dynamics like I just showed through. But then after the fact, instead of dispersing because they couldn't find any food, well, they ended up dispersing into the adjacent field where spider mites were first getting established and taking off. And it kept spider mites low the entire season. And so you can see, look at the predator-prey dynamics in these 2 figures here. In the June planting on the left, black line spider mites. So the spider mites get going, predators get established and take them out. And then they disperse into the July planting where they kept these spider mites low the entire time. So this is the case where a single release actually was benefiting 2 different crops. That's sort of doubling the economic situation. And we were also curious about how important the rearing source would be for predators. There was one previous publication on this in a greenhouse study that showed that when you took the same predators and you put them on, you either kept them on beans or adapted them to tomato for several generations, that those adapted on the tomato seemed to perform better, but it was a pretty limited study. And so we wanted to expand on this a bit. So as I said, commercial suppliers tend to rear their Persimilis specifically on bean plants. And so we compared it to the ones that we were rearing on tomato plants. So just like before, we went out into the field with either transplants of tomato covered in Persimilis or the shaker bottles that the companies provide. And then we shook those out onto tomato plants with spider mites to better control for that variable. And because these predators are so mobile, we tried to do this in a small plot study, and it was not very successful. Is that what we did here is we used a very large plots on in a grower field, split into replicated blocks like we would do in a typical experimental design. Each one of these plots is about half an acre. So it's way bigger than we could ever do on an experiment station. And so what we did was we either released bean adapted Persimilis, tomato adapted, or no predators at all. And we released 300 in the middle of each plot to give plenty of buffer area. And then we tracked those dynamics. And these figures here, instead of showing you the dynamics over time, this is sort of this is using total seasonal total cumulative mite days, which is more of a cumulative score of pest pressure. So it kind of encapsulates the whole season together into one figure. And you can see that in regards to spider mite suppression, the tomato adapted ones performed better than the bean adapted. And those were both better than doing no predators. So no surprise there. And Persimilis wise, we ended up finding a lot more Persimilis in the bean adapted plots, but that's because there was a lot more spider mites to begin with. And so the overall populations got a lot larger there, but the tomato adapted Persimilis were the most effective overall at suppressing spider mites and kept them low the entire season. And so this raises the question of why there might be differences among these strains. You could have differential performance that may be due to the physical environment. That would be the trichomes that are non-glandular, just physical, and they kind of slow insects down, and sometimes they can stab them and puncture the cuticle. You have the chemical environment, both the chemistries that are the plant chemicals that are released from the trichomes, or the ones that are inside the plant itself indirectly through the spider mite being fed on. So going to the predator that way. There could be differences there in these predator strains. They may also vary in their susceptibility to pesticides. So remember, this was a conventional tomato field where there were fungicides going out. There were other insecticides going out for stink bugs and herbicides. And so they may vary in that degree. And it's also unknown whether any of these differences would be due to physiological adaptation or to selection, where the populations are actually changing genetically over time. So these are answers we don't have yet. And regarding P. persimilis and the use of it is that, I've shown here that it can be highly effective. But persimilis and many other predators will never be successful if you use insecticides that are not compatible. So if you go in and use a broad spectrum insecticide like pyrethroids or organophosphates, that is going to kill the natural enemies and they'll never be successful. And so it's important to know which insecticides and other pesticides are compatible. And it is really important to know which miticides are compatible, because there may be a need that you need to be using miticides with predators, such as if the predators are introduced too late and the spider mites get too good of a head start. This especially is prone to happen under hot, dry conditions. And so you may need to go in with a miticide, knock the spider mites back a little bit, improve the ratio of predator to prey, and then from there, maybe the predators can take it. And so we have one publication on this here, evaluating the compatibility of acromite with persimilis in tomatoes. These two figures here are once again showing you the cumulative mite days, that seasonal overall calculation, and you can see that how this is for spider mites. And so the lowest one here is the persimilis acromite combination, showing that combination was the best at managing spider mites, and followed by persimilis acromite by itself in the control. And acromite, sometimes it can work and oftentimes can work really well, but there are other times it just doesn't work as well, possibly due to resistance issues. And so we've published a helpful guide online. You can find this on NC State's website to help you select compatible pesticides. We generated this chart ourselves from personal experience and research of papers and using some of the resources put out by these biocontrol companies. So this is the resource you can check out on the Wagenbach Lab website. And it's important to not just insecticides can be toxic to natural enemies, but there are certain fungicides and certain herbicides that have actually been shown to be toxic as well. And so it's kind of worth noting there. Dual Magnum has shown high toxicity to persimilis, for example. And again, we don't know, maybe among these different strains and populations, how much variation there is in susceptibility. In terms of rearing predators, I'll pick up kind of what Jim was saying. The process can be both easy, but also very difficult. And one thing that's important is you have to have separate locations for rearing these predators yourself. You really need 3 locations that are distinct: some place to grow plants that are clean and healthy and insect and mite free. And then you have an area that's used for rearing spider mites on the plants. And then you have another area that's then used for rearing the predators. And in fact, it can be most difficult to keep the predators out of your spider mite rearing area. And the predator area, it is important you'd want there to be the more optimal conditions for the predator development, so higher temperatures and higher humidity. It's kind of what it looks like here. Ideally, what you do is when the plants are still pretty small, you'd have a bunch of trays of tomatoes. You would inoculate those trays with spider mites, and then you would give it typical summertime temperatures. Will take 3 to 4 weeks for those populations to get really high. You want the spider mites to be as high as possible before predators are introduced. In this way, it maximizes the predator output. And so if you put predators on the tomato plants and you let them go for 3 or 4 weeks until you start to see webbing, that's a great time to add predators. And so then you would add the predators to those plants. You would wait another 3 to 4 weeks, and that is when you would have peak predator numbers, and that's when they would be ready to release in the field. So we've had really good success using this. When you get the system down, it's not very difficult. One person can do it with minimal space, although you need separate spaces. And an example for each batch, we would have 360 tomato seedlings. And we were able to produce up to 100,000 Persimilis. That's 300 predator per plant. And so if you were just releasing these in 10% of fields, that could service up to 50 acres of tomatoes. And that's not counting staggering tomato plants next to each other. So we had really good success with this. It can be a little tricky, but once you get it down, you can produce a lot of predators this way. And as Jim also mentioned before, is that we have these naturalized resident populations of Persimilis that are not just in North Carolina, but in many places up and down the East Coast. We study them closely in the Piedmont region of North Carolina, where they reoccur yearly, but oftentimes those populations by themselves aren't effective enough or reliable enough, which is why we did all these projects with releasing predators. And we had some projects ongoing about trying to understand the overwintering ecology, because that would help us understand how we can get these predators going earlier in the season and more abundantly. But it sort of just started. We don't have a whole lot of data yet on that. But really the goal would be to sort of modify the habitat in a way is that you can attract the predators to your fields earlier and in greater abundance and get the population going so that you wouldn't even have to go through the process of releasing yourself. And in North Carolina, we rarely, if ever, detected resident populations in the mountains, but in the Piedmont, we'd see them regularly. And so now I'm actually located in South Carolina, based out of Charleston, and I'm doing a lot of work in strawberries. And in strawberry fields in South Carolina, in both the around the coastal region and the Piedmont Sandhills region, over the last 2 years, I've detected Persimilis in most fields. They show up at some point, so it's very promising to enhance these predators in strawberries. And then that would tie into the research we're doing with tomatoes. And of course, many tomato growers often grow strawberries as well. We don't have, like I said, we don't have much data on this, but one approach we're using to both detect overwintering Persimilis and to potentially modify the habitat to increase their numbers would be using mint plants. There was some interesting work that came out of Japan that showed that mint plants are highly attractive to Persimilis, even when they're not infested with spider mites, which was a really striking result. And we have found this to be successful. We successfully, on many occasions, were able to attract and detect Persimilis all throughout the winter using these sentinel mint plants. And so perhaps in the future, if we put mint at the side of fields, it may attract them in greater abundance earlier. And just a final note here on this other predator at the end. And so we also had a research interest in this predatory mite, Amblyseius swirskyii. This predator is very different. It's a generalist predator, and so it feeds on a number of arthropod pests, mites, thrips, whiteflies, And this predator is of really high importance and of interest for managing Western flower thrips, which is a whole nother pest issue for another lecture. So there's a high interest in adapting this predator into tomatoes. It also feeds on pollen and nectar, and it's also a subtropical species. And we conducted many releases of this predator, and we were never able to recover them ever whatsoever. And we did some lab studies, and they're able to survive on leaves, but you try to put them in the field, and they just never survived. And we didn't get particularly far in this research aim, but unbeknownst to us, at the same time, there was a research group out of Spain working on this problem specifically with Swirsky. And this is a great paper in Biological Control published in 2021. And this paper showed that this is just some data showing that when you put Amblyssia swirsky, what is their predation rate when you do these tests? This figure on the left is just on leaves. So just tomato leaves by themselves, or tomato leaves versus pepper leaves, black versus white bars. And what you see is you don't see a difference. And so they're not really inhibited on leaves themselves, but when you do assays on stems where the trichome makeup are a lot different, is it significantly impacted dispersal and survival. And so it may be is that these predators could possibly do okay a little bit on tomato leaves, but they were never able to get them to do well in whole plants, and they can't move from leaf to leaf, and that pretty much just is going to void their use unless that can be overcome somehow. So some interesting work to check out of that research group. Overall conclusions is that this persimilis approach can be an excellent biological strategy for spider mites and tomatoes. But we need to improve the reliability a bit more to further improve their economics and make it even more compelling to use. And then a lot more work left on enhancing the resident populations. And of course, many people to thank, funding sources and other sources here in North Carolina. At that point, Jim and I will leave it open to questions. And I couldn't resist sharing this video we took of persimilis just zooming around spider mites on a strawberry leaf, actually.
Doesn't look like that guy's very hungry. Oh, he just got one.
It was actually too distracted by the fact that we had just put him on the leaf. We should have waited 24 hours for it to acclimate, but it wasn't happy about being confined to a small tanglefoot ring.
Thanks, everybody. I just wanted to start our question and answer session. Also, you can find information on this project, including some of the resources that were mentioned by Tom on the eOrganic website, and I'll just share the link to that in just a moment. It's on the eOrganic website at eorganic.info/biocontrol. That's the website for this project. And we'd like to acknowledge the NIFA, or Organic Research and Extension Initiative, for funding this project. And so with that, I'm just going to move on to the questions here. We have a few questions coming in. It's on what impact can the use of plant extract repellents have on predatory insects? So we'll start with that.
I can comment initially, and then maybe Tom can take off. There's been a fair amount of work looking at post-plant induced volatiles to attract predator insects, and I guess probably the most common one is methyl salicylate, which is attractive to a lot of different insects, and people have used sachets of that to bring predators into areas. I've seen a lot of work where you can attract predators with those products, but I haven't seen much in the literature in terms of actual impact in terms of reducing a pest population in a field setting. There's actually one that was pretty cool in a greenhouse setting, and they showed really good success in moving predators around in a greenhouse spot to hot spots using methyl salicylate like that. But I don't know if you have more information on that, Tom.
No, but another way, I'm not 100% clear on the question, but it may be, so the use of plant extract repellents, that could also possibly be in reference to some of the plant extract repellents, like maybe oils. Some of those are common in organic insecticides. If it was about that, by chance, those could have an impact on the predatory insects. They're not going to be necessarily specific to spider mites. And so I don't know of any existing research on if that's maybe what was being referred to, some of the essential oils and those products. I could see that being possible, but yeah, I'm not sure.
Okay, here's a question from Ben here. From the time of the first acaricide application to the first Persimilis release in your study, can an order for fresh Persimilis be placed and shipped? Another way to ask, how fast can Persimilis ship?
Go for it, Tom. You know that.
That probably depends on the company to some extent. The companies, you can work with a number of them. And if you have a good relationship, they can sometimes, Persimilis specifically, I think they can get them to you with a pretty quick turnaround. Sometimes you order it, they ship them the next day, it's overnight, so you could have them in 2 to 3 days, but it may be a week or maybe more. I've had one grower that tried to order it and there was a backlog, and you just never know these days, in the post-COVID world here with supply chain issues. But you would want to work with the companies if you're going to be purchasing them and understanding what the timeline would be.
Okay, let's see, another question here. Are there opportunities for bean-reared Persimilis facilities to modify their production to use tomatoes?
What do you think, Jim?
I'm not aware of companies doing that. Because there's not always a huge demand for spider mite or for Persimilis on tomatoes, I suspect. But
or there could be one day,
yeah, there could be.
But,
That's something to be interesting to bring up with some of our industry contacts. I don't have an answer for that.
Yeah, I mean, my understanding of why they use beans in the first place is because beans, actually anyone that does spider mite research knows that beans are probably the easiest plant to grow and to use and to produce lots of spider mite numbers. And so it's likely just related that spider mites are, in order to rear Persimilis, with one caveat, but in order to rear Persimilis, you have to have spider mites. And so you got to be really good at producing spider mites. And beans make it really easy to produce spider mites in a large abundance. And I believe that's really why they use it right now. But I don't see why, if there wasn't a big demand for tomato adapted Persimilis, and if they believed our results, then why someone wouldn't pick it up.
Great. I just wanted to say there was a question that you already typed the answer to, but others might be interested. Ko asked, can Persimilis overwinter in central New York in zone 4 to 5? And Tom had typed in, unfortunately, probably not, unless there are greenhouses or other places for them to reside. So yeah, thanks for that.
Maybe Jim, no, someone else that was in Jim's lab, David Owens in Delaware, has found, Jim, maybe you can, I don't know much about it, you could touch on that, but my general impression is central New York, it's pretty darn cold.
Yeah, I think once you get below, if you get temperatures below, below -5 degrees at any point in time, they don't have a very high tolerance for that type of environment at all. And you're going to get below zero temperatures in upstate New York. That's going to knock off just about everything. But in a lot of areas in the eastern United States, particularly along the coast, I'm sure all the way from Florida, and as Tom indicated, David Owens, who's an extension entomologist in Delaware, he's picking up resident populations of them in where they grow cucurbits up that way. So in a lot of areas in this mid-Atlantic area, or in the mid-Atlantic part of the country, you can grow them. We're becoming more of a Mediterranean type climate, I think, with climate change around here, and it overwinters very well in the Mediterranean area of Europe. So
Okay, yeah, and sort of a follow-up to Kayo's question here from Amara. Would you expect Persimmons to be able to build up a population and spread throughout a field during the summer in central New York? Or perhaps to put it another way, what minimum summer temperatures would you expect Persimmons to need in order to multiply and spread in a field?
Would be the minimum temperature. I see no reason why they couldn't take care of spread throughout a field in New York, in upstate New York. Gets pretty warm there in the summer. Yeah, I'm sure there's a lot of days in the 80s, and they'll do well in the 70s and 80s as well. One thing I should, and Tom, there's usually, from the time you release predators until you really start to pick them up in your sampling, there's about a 3-week lag.
Mhm.
And so you're not going to really, you might not think you're having success, but they're just got at such a low level initially, you're not picking up in your sampling. And then it's usually about a 2 or 3 week period after that where the spider mite population crashes. That's under the best situations. And I can say that we've done a lot of releases over the years, and we always show data that works. But a lot of times, for some reason, and I can never explain it, we might release them and they don't seem to take. And my only feeling is that it's got to be related to pesticides, but there might be something else going on there too. Any ideas, Tom?
Yeah, I'll touch on that. There's a lot of factors that are important for their success. A lot of things could go wrong. I'm working a lot with Persimmons in strawberries in South Carolina right now, and strawberries are a little different story because it's earlier in the season, and the spider mites can be problematic while it's still cooler out. And I've been doing releases in strawberries, trying to modify this sort of inoculative type approach. And in some fields, either a natural population or the ones released, they get established and they seem to do really well, albeit at a slower pace than they would in summer tomatoes. But in other fields, I had one organic farmer this year, 20 acres of organic strawberries. They released a lot of Persimmons, and we didn't pick them up at all until, it took 5 weeks and we found one. And so they didn't do anything despite lots of spider mites. And so that was an organic operation. So there really isn't that harsh of chemistries in that case. And that is, it's perplexing, these cases. We don't really know exactly why they don't do well. We know they won't do well if you go in and spray pyrethroid. That we know that much reliably. But there are other times that we're still trying to figure out. That was at the end there, improving reliability. We've had a lot of good successes, but it's not 100%. No approach is 100%, but
I just want to mention also that if you are interested in learning more about rearing them, Jim and Tom did another webinar a couple of years ago where they talked about that in more detail. So you can find that on the eOrganic YouTube channel and also on the eOrganic website under webinars by eOrganic, and it's in there in our archive there. So if you're interested in going into more depth about that, you can look that one up. Okay, so here's another question. You talked about this a little bit before, but how many two-spotted spider mites would indicate an issue when you sample the 10 leaflets?
Once you get to about, if you get into the point of 10 mites per leaflet, so that would be 100 mites on 10 leaflets. Something needs to be done if there's no predators out there, because they'll continue to rise. And that's the point at which that's kind of critical point when they start to inflict injury. And if they get much higher than that, they're really going to be difficult to oftentimes control, because so I would say if you get to 80 to 100 mites per 10 leaflets, you better think about doing something. But that's too late if you're interested in biological control.
Okay.
And for biological control, what you're going to want to do is as soon as you pick up the first mite, essentially in the field, you got to think about releasing the predators.
But you
Okay.
can't release them before there's any spider mites because they got to have, it's their only food source. So there also needs to be, so you got to be scouting. Entomologists always tell folks, you got to be out there. You got to be looking for pests. You got to be looking for diseases.
Yeah. Okay. Yeah. And there's some really great information on Jim's website in the Walgenbach lab with great photos of different pests and predators. So I definitely encourage everyone to go look at that for help with identification. For homeowners or organic gardeners, is Organicide effective on two-spotted spider mite? Active ingredients include sesame oil, fish oil, soy lecithin, and potassium sorbate. Have you tried that at all?
I've tried. Go ahead, Tom.
I have no experience with those particular oils, so I can't speak to that.
I've tried some things in the past. I think it was like a couple of different, I can't remember. Garlic oil was one time. I've never had a lot of success with those.
Okay.
The only one I've had good success with is like Venerate and sulfur and some of these oil products, but not specifically those like garlic oil or the other type of oils. Matter of fact, the only oil I've had good success with is emulsified oil, the one that's called Tritech.
Okay. Are there any relatives of Persimolis in the Northeast U.S. That is known or studied?
There's another species in the genus Phytocelus macropilus, but I'm not sure if it's detected up there at all or is present. It's not really produced by the companies much, so I'm not. I don't think so. Is my answer to that.
Okay.
Unless Jim knows otherwise.
Okay. Is it better to spread the mites along a row or across rows at one end? Or in other words, what's the best way to apply the mites?
Logically, you would think they're going to move down a row a lot easier than they would across rows. But we seem to have quite a bit of success with those things moving across rows. They're good walkers and they're fairly fast, so they can, if they're motivated to leave a plant to go next door to get some mites, they can do it.
Yeah, I think in some studies of comparing different phytoceid predatory mites, that Persimolis was it has the fastest walking speed. They use little trackers under a microscope, and Persimolis is like the fastest walker. And so yeah, it's mobile, as we alluded to multiple times. But they do spread. Dispersal, it probably differs among different cropping systems. State tomatoes are a little bit higher up, and so for those that are going to be dispersing by wind, which a lot of mites do as well, we probably see good dispersal in tomato. I would not be surprised if dispersal rates were not as rapid in, say, strawberries, low growing.
Okay. Let's see. Where can we buy the predators?
I think as university employees, we can't endorse any specific.
Yeah, but I mean, can somebody just Google that and find a company that sells them? Is that?
What I'll say about that is that there's a handful of places that actually produce the predators and sell them, and then there's a lot of companies that are intermediaries. And whether that ends up making a difference at the end of the day, I'm not sure. I don't even know if there's been one or two studies done on that, but it's been so long ago that the data may not be relevant anymore because these biocontrol companies are changing a lot. But you can look on yeah, you can look online and maybe if you know other people that have bought them in the greenhouse industry and have had good success, you could talk to them and try it out. Yeah, but
But
so
you can Google it online and find reputable
yeah.
sources, no problem.
Okay. Let's see, I'm just scanning here to find if there's any other questions here. Let's see, I think there was one more here. Oh yeah, what can we feed for the reproduction of Persimolis?
Spider mites.
Yeah, you
That's
said
it.
that's all they eat, so
Yeah.
yeah.
If they don't have that, they starve to death.
There was this is something that Jim and I talked about doing some research on at some point, but there was just one study that was able to extend survival with some sucrose solutions, but primarily it's the only thing they're really consuming are spider mites. So maybe there's a role for certain supplementing nectar or sucrose in some kind of way, but spider mites, that's why they're and actually I should add, I just learned about this. One of the companies is now they have somehow discovered a way to find another artificial or not an artificial food source, but they have found another mite that they've been able to get Persimolis to feed on. So it changes their color. Actually, they are more brown instead of being red, and I'd be interested if that impacts their predation or survival in any way, but we don't know. But really, spider mites, yeah.
Okay, yeah, I just shared a name of an organization that Elaine very nicely typed into the chat here. I guess a source of finding biocontrol producers is the Association of Natural Biocontrol Producers, called the ANBP. So
Yeah, thanks, Elaine. I forgot about that. That's really a good source.
yeah, that's really helpful. Thank you. Okay, and I guess, yeah, maybe this answers Jane's question here. What are some things to look for to know if a company is reputable, or maybe to look for to avoid another company? Elaine just typed in the name of that organization, which some companies may be members of, so that might help.
When you buy predators, I know that if they give you a very specific list of things that you're supposed to do to ensure quality, because this is not just like a chemical in a bottle, and they expect you to follow those very closely. They want you to release them right away, among other things. The timing, I think they have some other guidelines, and you definitely want to, when you get them, even though mites are really small, you should try to pour some out on a white piece of paper, and you want mites that are moving around. Make sure they're still alive when you get them.
Good point. Okay, we have time for one more question here. What are folks using for stink bugs that won't hurt Persimolis or aureus?
Jim?
Unfortunately, what most people are using for stink bugs are pyrethroids, which are not good for Persimolis. Probably something that's some of the neonicotinoids, you can get things to survive quite well. Now, one is acetaminoprid. Acetaminoprid is it's not the greatest stink bug material in the world, but it's awfully safe to Phytocilius. And another one is thiamethoxam. I encourage our growers to use when we are releasing predators in there, and they seem to survive that quite well. So thiamethoxam, which is, there's only one product out there, and that's Actara. But in our experience, it only takes, I know a lot of the growers we work with in North Carolina, they only need to put something in for stink bug about once every 3 weeks or so during the period to avoid stink bug damage. So
okay, yeah, and we always add a disclaimer to all of our articles that if you're an organic producer, always check with your certifier before using any product, and also make sure it complies with the local and state regulations.
And I should point out, in terms of organically, there's not too many good stink bug options from an organic perspective. Azera might be one of the best, but that does have a pyrethrum in it.
Okay. Well, thank you both for this discussion, and also thanks to everyone for submitting all these great questions. And I just want to say one more time that this is recorded, and we're going to be posting that within the coming week on the eOrganic YouTube channel. You can find the other webinars by Tom and Jim there, as well as the other ones in this series that talk about some of the other pests and biological control insects that Jim and Tom talked about today. So thanks, everyone, and we'll see you back probably in the fall for more webinars.
Thanks so much for the question, for attending, everyone.